Hello students and welcome to EPG Pathshala. I am Dr. Sudhir Verma, Assistant Professor, Department of Zoology, Deen Dayal Upadhyay College, University of Delhi. Today we are going to talk on module Complement System from the paper Immunology. So students, let's see what we are going to learn in this module. We'll start with the introduction of complement system as to what complement system is, what it is made up of, how it actually acts as an interface between innate and adaptive immunity, etc. Starting with this thing, we'll move on to the history part, how it was actually discovered. Then we'll move on to the various functions, various physiological activities that it performs. We'll see the various norms and the rules, how these complement proteins are named. Then we'll move on to the pathways of complement system, various receptors and regulators that actually regulate this complement system. We'll move on to the diseases that complement deficiency leads to, various assay systems, how these deficiencies are detected. And lastly, we'll see how various microbes actually evade this complement system to survive. Moving on with the introduction part, the complement system defined as activity of blood serum that completes the action of antibodies. It was for the first time discovered by Jules Bordet in 1890 as a heat labile component of serum that complemented or augmented its bactericidal properties. Comprises of 30 soluble proteins so far which have been isolated, and together it constitutes approximately 10% of total serum proteins. Most of these proteins are synthesized by liver and remain in inactive form in the absence of infection. Evolutionary, if we see, complement system is ancient as adaptive immune system, as even worms and starfishes have also this system. It consists of primarily three different activation pathways, the classical one, alternative one, or the lectin pathways, which ultimately leads to a common terminal pathway that forms a membrane attack complex which subsequently lyses the target cells. Complement system broadly performs three types of functions. Host defense. It acts as an interface between two branches, innate and adaptive immunity, and the clearance of waste, which may include apoptotic cells and the immune complexes. Various receptors and regulators are involved in functioning of complement systems. Number of diseases have been reported which occur due to genetic or functional deficiency of various complement proteins. Still, there are a number of pathogens which actually evade these complement system, and they have evolved various strategies for that. Let us discuss these points in detail now. To start with, let's have a look at the history of discovery of complement system. Jules Bordet in 1890 in Pasteur Institute of Paris showed that sheep antiserum to bacterium Vibrio cholerae caused lysis, and heating the antiserum destroyed its bacteriolytic activity. But this ability to lyse the bacteria restored to heated serum by adding fresh serum that contained no antibodies against the bacteria, and by itself was unable to kill it. Bordet reasoned that bacteriolytic activity requires two substances, that is, specific antibody and heat-sensitive component. This heat labile component was named as alexins by Bordet. Later, Paul Ehrlich coined the term complement for the same component. Bordet was honored with Nobel Prize in Physiology and Medicine in the year 1919 for his discovery of complement-mediated bacteriolysis. Moving on to biological activities of complement systems. The biological activity of complement system reach far beyond these original observations of bacterial lysis. After initial activation, the complement components interact in a regulated cascade and carry out a number of basic functions such as host defense by lysis of pathogenic bacteria and cells, opsonization, which promotes phagocytosis of particulate antigens, activation of leukocytes, augmentation of antibody response and enhancement of immunological memory, immune clearance by clearing antigen-antibody complexes from the circulation, and clearance of apoptotic cells, etc. Here you can visualize some of the key functions of complement system that it performs. Now let's have a look at the international code for naming the components of complement system. The complement factors are named by various methods. To start with, the complement factors can be named either by the numerals such as C1, C2, C3, etc. or by letter symbols such as factor B, factor D, or also by their trivial names such as decay accelerating factor, that is DAF. As we mentioned earlier, these proteins are present in inactive or zymogen form and become active upon cleavage. The peptide fragments which generate upon cleavage are denoted by small letters. The smaller fragment is named as A and the larger one is called as B. The C2 protein is an exception to this rule, means C2A is a larger component and C2B is the smaller one. The larger fragment binds to target near activation site and the smaller fragment diffuses out and initiates the localized inflammatory response. The complement fragments interact with each other and make complexes. The complexes, which possess catalytic or enzymatic activity, are denoted by an overhead bar. Now, keeping these rules in mind, let's move further to different activation pathways of complement system. The overall cascade can be divided into an early activation steps and a terminal membrane attack complex formation. The early steps of complement activation can follow either classical, alternate, or lectin pathway. All of them culminate in the formation of C5, that is, the cleavage product of C5, that is C5b. The terminal pathway is common to all. Let's move on to the classical pathway. The classical pathway of complement activation starts when the complement C1 recognizes a microbial surface directly or indirectly via antibody bound to pathogen. C1 complement is a complex made up of a large subunit C1q, which senses the pathogen, and two zymogenic subunits C1r and C1s that act as serine proteases. The pathogenic recognition activity of C1 lies within the globular domain of C1q. When these globular heads interact with a particular ligand, a conformational change is caused in C1r and C1s. The resulting active form of C1r then cleaves C1s to generate active serine proteases. C1s acts on two substrates, that is C4 and C2. C4 is a glycoprotein that consists of three polypeptide chains, that is alpha, beta, and gamma. C1 hydrolyzes the C4a, that is small fragment of C4, from amino terminal of alpha polypeptide chain and exposes a binding site on remaining C4b component. C2 attaches itself to the exposed binding site on C4b and is cleaved by C1s to C2a and C2b. This smaller fragment C2b, as mentioned earlier, C2 is the exception in nomenclature, C2b being smaller diffuses away and C2a attaches itself to target surface along with C4b. The resulting C4b to A complex is called as classical C3 convertase enzyme, as it activates C3 from its zymogenic form to active form. C3 complement consists of two polypeptide chains, alpha and beta. C3 convertase hydrolyzes C3a from amino terminal of alpha chain of C3 and generates C3b. The C3b thus generated binds to C4b to A complex, giving rise to trimolecular C4b to A 3b complex, which is termed as C5 convertase. The C5 is cleaved by C5 convertase to C5a that diffuses away and C5b, which binds to C6 and starts the membrane attack complex formation. Various proteins involved in the classical pathway of complement activation are listed here. Moving on to the next pathway, that is alternate pathway. The alternative pathway is antibody-independent innate immune system component. It is activated by foreign cell surface and does not require antibody to initiate. In alternative pathway, serum C3 containing an unstable thioester bond undergoes slow, spontaneous hydrolysis to generate C3a and C3b. The C3b component cannot differentiate host surface from the foreign surfaces, but because of high sialic acid present on host surface, it rapidly deactivates and does not lead to further cascade. Whereas on foreign surface such as bacteria, yeast and virus, low sialic acid is present and thus C3b remains active. C3b then binds factor B by a magnesium-dependent bond which provides a surface for serum protein D to bind. Factor D cleaves factor B, releasing BA. Remaining complex C3b possesses C3 convertase activity that is very unstable. Another factor, that is properdin, stabilizes this complex. The C3 convertase further activates C3 to generate more C3b, leading to formation of C5 convertase, that is C3b, B, B, 3b. The C5 is cleaved by C5 convertase to C5a that diffuses away and C5b which binds to C6 and starts the membrane attack complex formation. Here you can see the various proteins involved in alternative pathway of complement activation. Lectin pathway is also antibody-independent innate immune system component. Lectins are the proteins that bind to carbohydrates. This pathway initiates with the binding of mannose binding lectin, that is MBL, to mannose residues on surface of microorganisms. MBL is an acute phase protein analogous to C1q of classical pathway. MBL binding leads to binding of MBL associated serine protease, that is MASP, which further cleaves and activates C4. MASP is structurally similar to C1R and C1s. Rest of the mechanism is similar to classical pathway for generating C3 and C5 convertase, leading to membrane attack complex formation. Here you can see the various proteins involved in lectin pathway of complement activation. C5 convertase, upon activation in all the three pathways, cleaves C5 into C5a and C5b. C5a diffuses away, whereas C5b binds to the target surface. C5b, being very labile, is stabilized by C6. Upon binding of C7, C5b6-7 complex undergoes hydrophilic amphiphilic structural transition, exposing hydrophobic regions that serve as binding sites for membrane phospholipids. If this reaction takes place on the target surface, C5b6-7 inserts into phospholipid bilayer, whereas if it occurs on immune complexes, then the released C5b6-7 complex enters into the membrane of nearby cells, causing innocent bystander lysis, that is an autoimmune damage. Next, C8 binds to C5b6-7, which creates a small pore of approximate 10 angstrom. 10 to 17 molecules of C9 polymerize around C5b6-7-8 complex and increase the pore size from 10 angstrom to around 100 angstrom. This pore causes osmotic instability in the target cell because of water influx and loss of electrolytes, ultimately leading to cell lysis. Various proteins involved in the terminal pathway of complement activation are listed here. Here you can see a comparison of three activation pathways in terms of the precursor, the activating protease, composition of C3 and C5 convertase, the C5 binding component, activation, and the component of innate or adaptive immunity. Moving on to the receptors for complement proteins. The biological activity of complement system depends on their binding to various receptors present on different cell types. These receptors not only mediate biological function of complement proteins but also regulate them by inactivating the activated form of complement proteins upon binding. Some of the major receptors and the cells which they are present in, along with their ligands and functions, are shown here. For example, in erythrocytes, B cells and macrophages, there is a receptor called CR1. This receptor binds the ligands C3b and C4b, and it blocks the formation of C3 convertase. Similarly, different cell types have different receptors which have specificity for the ligands and carry out particular functions. The regulation of complement system. Being non-specific in nature, elaborate regulatory system has evolved to restrict this non-specific activity of complement system on the host cells. A general way of regulation is achieved by labile nature of all the complement components, which undergo spontaneous inactivation on diffusing away from the target cells. A number of regulatory proteins also serve as a regulator of complement system as shown in the table. Many of these proteins are encoded at a single location on chromosome 1 in humans and known as regulators of complement activation, that is RCA gene cluster. The regulatory proteins operate at all cell types and levels including classical, alternative, lectin and terminal pathway, and also at the effector level. Here you can see a list of various proteins, the pathway where they are involved, and the different immunological functions that they carry out. The deficiency of complement components can lead to various disorders. These disorders are generally complement deficiencies which are rare in nature, but some of them are very common in specific racial groups. Some of the common impacts or diseases arising due to complement deficiency are deficiency of classical pathway. The deficiency of any of the classical pathway component, that is C1, C3, C4, C2, predisposes an individual to a condition similar to systemic lupus erythematosus or SLE. In SLE, immune complexes, that is antigen-antibody complexes, gets deposited in skin, kidney, blood vessels, etc. Deficiency of alternative pathway components, that is factor B or factor D, reduces the efficiency of opsonization of individuals. It leads to recurrent bacterial infections in the affected people. The deficiency in lectin or MBL pathway. MBL deficiency in infants make them susceptible to bacterial infections. But as the age progresses, this susceptibility disappears as other immune mechanisms mature. In adults, MBL deficiency doesn't lead to any serious consequences unless accompanied with other immunosuppression or immunodeficiency. Deficiency of terminal pathway components, that is C5, 6, 7, 8 or 9, predisposes the person to gram-negative bacteria infection, particularly Neisseria genus. Gonorrhea and meningococcal meningitis are common diseases caused by Neisseria. The deficiency of C1 inhibitor, which is a classical pathway regulator, leads to hereditary angioedema, which is an autosomal dominant disease. Deficiency of properdin, which is a stabilizer of alternative pathway C3 convertase, shows X-linked inheritance in its deficiency. Thus, properdin deficiency is exclusive in males. Males deficient in properdin shows meningococcal meningitis. Now let's have a look at the assay systems that we can utilize to detect various complement deficiencies. It may include the global assays for identification of lack of pathway function, either hemolytic assays or enzyme-linked immunosorbent assays with IgM, menin or LPS. Created plates is used for such analysis. The total functional activity or the functional activity of single component can be performed in such assays. So, any sort of defect can be detected directly if there is a structural deficiency. Another method that we can utilize is the protein, chemical cytofluorometric and the functional assays for individual component and the regulators, irrespective of their functional activities. These methods may include the radial immunodiffusion methods, nephelometry, and also again the enzyme-linked immunosorbent assays. So in these ways, both the structural as well as the functional deficiencies can be detected easily in the research labs. Another very prominent method is molecular analysis. These molecular techniques can be used to identify genetic alterations. Now these genetic alterations can be mutations, it can be polymorphism in various components of the complement system, and they can be studied by method of genetic mapping. Though complement system is very effective in providing immunity against various pathogens, but microbes also have evolved a number of strategies to breach this system. Complement system is more effective against gram-negative bacteria as compared to the gram-positive ones. A few gram-negative and most gram-positive bacteria adopt different survival strategies by fooling our complement system. For example, the gram-negative bacteria, because of presence of polysaccharide chain in cell wall, the proteins in the outer membrane, and the presence of elastase enzymes, have prevention of MAC to insert in their membrane, or inactivation of anaphylatoxins C3A and C5A. The pathogenic forms of E. coli, Salmonella, Neisseria, and Pseudomonas are the common examples. Among gram-positive bacteria, the presence of bacterial capsule and peptidoglycan in cell wall prevents the MAC insertion and the physical barrier between C3b and CR1. The Streptococcus are common examples among these categories. Among other mechanisms, inhibition of complement cascade because of molecular mimics of complement regulators is commonly found in herpes and vaccinia. So students, let us now summarize what we have learned in this module so far. We have seen that complement system is a component of our immune system which comprises of almost 30 soluble proteins. Most of these proteins are synthesized in liver, though other organs are also reported. Most of these proteins are present in inactive form, which we also know as zymogenic form. They remain inactive until there is no infection. The moment there is an infection detected, there is a cascade starting of all these components, which ultimately leads to formation of what we have seen, the membrane attack complex. As far as discovery we saw, the Jules Bordet for the first time identified these complement systems. It was identified as a heat labile component of serum. And this heat labile component complements or augments the bactericidal properties of antibodies. Though Jules Bordet named them as alexins, the Paul Ehrlich named them as complement, which we know them as of now. The complement system is the major component of our innate immune system. Whenever it is encountered with pathogens, which can be exogenous as well as the endogenous, which enter our body after breaching the various physical barriers and the antimicrobial defenses. It's not only the part of innate immune system, it provides a link between the two branches of immunology, which we know as innate immunity and the adaptive immunity. As far as activation is concerned, 3 different pathways are reported. These pathways are differentiated on the basis of what is the initiating agent. What are the proteins which are actually involved in these pathways? Though the terminal pathway is same, but activation can be differentiated in 3 pathways as the classical pathway, the alternative pathway, and the lectin pathway. The ultimate result of all these pathways is the lysis of target cell by the formation of what we call as membrane attack complex. As far as the functioning of complement system is concerned, all the functions can be broadly categorized in 3 categories, which can be the host defense against various infections. These infections again can be exogenous as well as some endogenous defects also. It provides a link or interface between innate immunity and the adaptive immunity. Besides these, another important function that they perform is the clearance. Antibodies which bound to antigen, they form a complex of antigen-antibody which we call as immune complex. Besides this immune complex, we do have number of apoptotic cells which has to be cleared up from the body. Complement system actually provides a support for clearing of these waste. The biological activities of complement system are governed by binding of these complement proteins or components and the fragments with various receptors. These receptors can be present on different cells. Additionally, these receptors regulate the complement activity. Various regulatory proteins are present in our body that keep a check on the activity of complement system so that they can avoid any sort of damage to the host itself. So it's a preventive strategies that we have. Number of genetic or functional deficiency of different complement proteins, maybe the structural deficiencies or the functional deficiencies. They result in number of disorders or diseases, which illustrates the homeostatic role of complement system. A number of diagnostic methods have been developed in past which are available to detect the overall complement deficiency or any component deficiency of any of these complement pathways. Now, these diagnostic methods are widely utilized to know which component or which pathway is actually deficient in a person. Besides being a non-specific mechanism and having so much regulation, the bacteria or the pathogens which are coming from outside, they have also evolved a number of strategies to breach this system as well. Particularly the gram-positive bacteria and some of the gram-negative bacteria along with viruses are among these smart evaders. They have developed strategies. Now these strategies can be the molecular mimics, can be the structural strategies which allow them to evade this system. So with this, thank you so much.